Hello, my name is Hunter Wagner, and in this video I'm going to be demonstrating duplicate recessive epistasis through the example of the Eldar from Warhammer 40k. So Warhammer 40k, or Warhammer 40,000, is a futuristic dystopian collection of books. And in this collection of books there exists a race called Eldar, which are the equivalent of space elves, and this race got mutated at the hands of a god, which formed the offshoot of the Dark Eldar, which are a more violently predisposed group of individuals of the Eldar, and do differ genetically from the Eldar. So what we see here is the duplicate recessive epistatic pathway. Now, it sounds difficult, but it's really pretty simple. All epistasis is the masking of the showing of a trait through genetics. So in duplicate recessive, if we have either a recessive A or a recessive B, then we do not show the Dark Eldar trait. However, if we have both a dominant A and a dominant B, we have the coding for both enzyme A, which progresses us to a intermediate form of the Eldar, and enzyme B, which progresses us from the intermediate to the Dark Eldar. So here's what happens in epistasis when we get a dihybrid cross, a cross between two individuals who are both hybrid in both genes. So these are the general crosses of all the gamete types that could possibly be produced by each individual. And on the bottom here, I just put totals of all your genotypes that you could possibly get out of 16. Now, if we take those two and we run them through that pathway I showed you earlier, and you'd see that 7 out of the 16 had a dominant A and a dominant B, while 9 out of the 16 had at least a recessive A or a recessive B to stop them from progressing to the Dark Eldar. Let's say we don't have two individuals who are heterozygous in both loci. Let's take a Dark Eldar individual and cross it with a Old Eldar individual. And if we do that, we'll get this table down here. And you see we get a perfect split of 4 in each category. However, this doesn't mean we have a perfect split in our displayed traits. As we can see here, we have 4 individuals, or 25%, with at least one recessive gene, and we have 75%, or 12 out of 16, with 2 dominant alleles.